Welcome back to the second part of our exploration into the body-on-chip research. Today we delve into the world of neurovascularization, the integration of blood vessels and nerves into our miniature body models. Neurovascularization is the process of mimicking the intricate networks of blood vessels and nerves found within our bodies. To model and to study the interactions between neurons and blood vessels, we have used two cell types: human umbilical vein endothelial cells, HUVECs, and neurons. We have combined the two cell types on a miniature platform, a chip. Why, you may ask? Well, let's hear it from the experts themselves. Welcome to the studio, Lotta and Alma. Thank you. Can you tell me, how did the project with nerves and blood vessels begin?
I started my PhD in 2019, and quite quickly we realized with my colleague Anastasia that we are both aiming to have these perfusible networks in our first study. So we decided to combine our forces. We managed to get these beautiful, small, tiny blood vessels that actually could transport particles within.
Yeah, and after Alma and others had formed this beautiful vascular model, we started to have this idea that we should actually integrate also the neuronal network into the same model so that we would even better mimic the situation that we actually have in our bodies. It wasn't maybe that straightforward as we were hoping at the beginning, but we managed to do that, and eventually we had this model with both neuronal and vascular networks together.
Could you tell us why integrating blood vessels and nerves into our body-on-chip models is needed for advancing medical research?
We have neurons and blood vessels in basically almost all of our tissues. Due to this central role in the functioning of our tissues, also the dysfunctions in these networks affect a lot of diseases, for example. In brain cancer, the tumor cells interact with both neurons and the blood vessels, and with this approach, we hope to understand the progression better and hopefully even find some novel ways to treat this disease.
We need to have all the pieces of the puzzle, so to say, to see the whole image and all the effects.
We have mentioned chips quite many times in episode 1. Can you explain how these chips are made?
We've been actually both working with custom-made chips that you really design and manufacture in the university, but you can also buy them online. What we do is that we have the cells, we need the blood vessel forming cells, and then we need the blood vessel supporting cells. And we mix those into this gel-like structure, giving the binding sites and something for the cells to attach. And then this tiny drop, since we have the cells and we have the hydrogel, we put it in these devices. In there, the cells they have the right environment, and then they know what to do.
Yeah, exactly. Essentially, the cells do actually all the work, you could say even. So we mix all the cell types together, and then they self-assemble to form these beautiful networks there.
Yeah.
Were there any surprises along the way?
Oh, I think working as a researcher every day is a surprise. You of course have protocols, but when you go to the lab, the outcome can be something that is completely unexpected. And I think that is the beauty in science, that we are exploring the unknown and discovering things that no one actually knows.
I have to say that also you really face these kind of positive surprises. So you are really blown away by the results that you obtain. We all were really surprised that neurons support the blood vessels and vice versa. So that was really surprising to see.
Thanks for the wonderful conversation here today.
Thank you.
In the grand scheme of the body-on-chip project, neurovascularization is just one important puzzle piece. So stay tuned on our video series as we will discuss the heart and liver modeling in the next episodes. Thanks for watching.